Now, I'm going to introduce latent bone modeling approach to estimate plutonium activity concentration in human skeleton. George's presentation compared simple average and mass-weighted average method, but the following 4 presentations of the USTR bone series deal with latent bone modeling. So first, what is a latent bone? Latent bone is a latent variable that is not directly measured, but is rather inferred through a mathematical model from other variables that are measured. In other words, latent bone plutonium activity concentration is not directly measured, but is rather inferred from measured bone concentrations. Mathematical models that aim to explain observed variables in terms of latent variables are called latent variable models, and in our case, called latent bone models. My next few slides are about background information and the USTR database. But thanks to George's presentation before me, I will not repeat them. So I'll skip why plutonium is in the skeleton. And the USTR whole body bone data set contains 19 whole body cases and 90 bones. USTR partial body bone data set contains 232 partial body cases with 2 to 8 analyzed bone samples. The most commonly collected 3 bones are rib, sternum, and the vertebral body. For more than 3 decades, a practical research question has been how to estimate total skeleton plutonium concentrations of partial body cases with a limited number of bones. There are a number of published USTR studies dealing with this question since 1988, and the recent publication was in 2019. The linear regression approach was used in several studies based on assumption that there exists a linear relationship between plutonium concentrations of a bone and the whole skeleton. 3 types of models were attempted using whole body cases: single bone linear model, group bone linear model, which uses the average bone concentrations of selected bones, and multiple linear model. In general, the multiple linear model is better than group bone linear model, and the group bone linear model is better than single bone linear model. However, there are 2 problems with the multiple linear regression approach using USTR bone data. The first issue is multicollinearity. Individual bone concentrations are highly correlated with each other. For example, this is the correlation matrix for 6 commonly collected bone at autopsy in USTR. The correlation coefficients are all above 0.9. One of the assumptions for a multiple linear regression model is that independent variables should be independent from one another. Such high correlations between bone concentrations are problematic and will result in imprecise and unstable estimates of the model parameters. The second limitation is the small sample size. Phillips' study in 2003 used only 8 whole body cases available at the time in the multiple linear model with 2 to 5 bones as independent variables. For 8 cases with 2 bones, the case to variable ratio is 4. And with 5 bones, the ratio is less than 2. So the case to variable ratios were too low, resulting in the model overfitting. Even with the 19 whole body cases available now, it is inappropriate to include 4 or more bones in a multiple linear regression model. To overcome the 2 limitations of multicollinearity and the small sample size, a statistical technique called principal component analysis is applied. For the interest of time, I will not discuss the technical details of principal component analysis in the presentation, but the objective of principal component analysis is to reduce the number of original independent variables by finding fewer principal components that are preserving as much variability or information as possible in the original data. Principal components are linear functions of original variables and are orthogonal to each other. Principal component regression is a multiple linear regression analysis technique that is based on principal component analysis. In principal component regression, instead of regressing the dependent variable on the independent variable directly, principal components are used as a regressor. Typically, only the first few components are used for regression. In our context, the principal component is called a latent bone, and the principal component regression is called latent bone modeling. The diagram shows the 2-step process of principal component regression. The first step, principal component analysis, PCA, and the second step, multiple linear regression, MLR. The original data matrix, C-bone, has 19 whole body cases as rows and 90 individual bones as columns. It is interesting to note that in principal component analysis, the number of rows can be less than the number of columns. Or in other words, the number of cases can be less than number of variables. After principal component analysis, the latent bone matrix has M rows and K columns, where K is the first few principal components or latent bones, and K is much less than N. As a matter of fact, K equals 1 or 2 in our models, which means that only 1 or 2 latent bones are used in the multiple linear regression. With 19 whole body cases and now only 1 or 2 independent variables, the case to variable ratio is acceptable. Now, I would like to present an example of latent bone modeling using the 3 most commonly collected bones: rib, sternum, and the vertebral body. The table contains the plutonium concentrations of the 3 most commonly collected bones from the 19 whole body cases. The first step, principal component analysis, produces the first latent bone concentrations, which are linear combination of the 3 bone concentrations. Note that the 3 bone concentrations are standardized to have a mean of 0 and a standard deviation of 1. The coefficients are called loadings or weights of bone concentrations. The weighted sum of 3 bone concentrations is called latent bone scores or latent bone concentrations. In this case, the first latent bone alone explains 98.4% of total variance. The second step is a simple linear regression using first latent bone as an independent variable. This table contains the data used for the linear regression. The total skeleton concentration can be estimated using these linear equations with adjusted R-square of 0.96. The figure shows the latent bone model predicted versus measured skeleton concentration, and the model fit is pretty good. In summary, this presentation introduces the latent bone model approach, which is the preferred method for estimating plutonium concentration in the total skeleton from a selected number of bones. Latent bone model for the most frequently collected 3 bones were described as an example. Thank you.